you'll be required to learn 2 components. You'll be required to learn the first component, which was taught at main campus, and then there's another component that's taught at 3rd year. But for us, since it's 6 years, medicine is 6 years, so you have to learn both components in 2nd year, meaning you have a lot of things to study. So you start from where you ended on your biology. You did your proteins, you did your lipids, you did your fatty acids, you did your water. So that's what will be your first component of biochemistry. And then you go deeper into the chemical reactions, how the body uses those things to do the metabolizing in the body. So I don't know if there are any questions before I can go on. Are people getting me? Do people have questions so far?
I think you can go on.
Okay, so yeah. So biochemistry, personally, I found it to be one of the challenging courses because from the word go, I never used to study because you have a lot of things. Yes, you have 7 courses, but you just need to divide your time. I remember being also told when I was in my 2nd year, being told it's a silent killer. It is because your CA will be composed online. You won't have any physical tests. You won't have any physical quizzes. Everything will be done online. So you do not take advantage of that by copying from your friends, not knowing what is happening, because your CA will be done online. So you have your friends to help you. You have groups to study with and help you, but do not neglect your personal studies. Do not neglect knowing what is happening. Do not depend on people. I'm not saying do not depend on them, but I'm saying do not depend on them 100%. In that, if you were told to write that paper alone, you wouldn't make it. So your CA will be made online. You have to write your CA, your test 1, 2, and 3. You have 3 tests. They'll all be online. And so make sure you perform very well. It's the CA that will actually rescue you and make you clear because it will be all online. So why would you risk it? Why would you do it alone? Do it with friends. Make sure you have people who can help you. Make sure you have seniors as friends. Do not totally depend on yourselves to help yourself. You need a mentor. You need, in as much as you are getting tuitions here, in as much as you are getting help, have someone as a 3rd year that you're looking up to who's coaching you. So in as much as you're getting this, make sure that you have direct connections with your seniors for guidance. How do I do this? How do I know this? You need that. So as you write your CAs online, make sure your goal is like having an 80% and above in every assessment that you'll be given. Every text that you have, make sure you have at least 80% and above because it's online. So they have a tendency of repeating questions. So make sure that you have all the data necessary to you. You can have them if you want. I can share with you. I do have some screenshots. So they might repeat the same questions even this year. So your tests, please make sure that you clear them. Make sure that you have at least 80 and above because that's the only 80 you have. You won't manage to have that 80 in the exam. So once having that, also make sure that you also understand. You also make sure that whatever assessment they give you from this tuition center, make sure you answer it. Make sure that you take the points, the key points that they tell you to take note of. You're taking note of them because that's what can assure you there are a lot of things in this course. You have a lot of things to know. You have a lot of processes, a lot of chemical reactions to know. If you thought you're running away from organic chemistry, it's also part of organic chemistry. So you have some elements of that. So make sure you just have time for biochemistry. Otherwise, it's not a difficult course. People clear it. Yeah, you can also do the same. Thank you. Unless there are any questions, I think that's it.
Thank you very much, Doc. So, someone just raised their hand. You can kindly unmute yourself and speak, please. Your mic is muted. Kindly unmute yourself and speak.
Okay. Yeah. My concern is just on the voice is too low. You can't hear. I can't get you so clear.
Oh, sorry, you couldn't hear me. Can you hear me now?
I'm, I'm hearing you, but your voice is too low.
Okay, can you hear me now?
I think it's just on his end. Otherwise, I can hear you.
Yes, it's just in.
Okay. Others got what I said, right?
Mukata, kindly turn off your video. I think it's interrupting with audio connection. Kindly turn off your video. All right. So I'm sure you guys have gotten what Annie was telling you, and she may have to leave now because we are about to start class. Annie, thank you very much um, for your words. I'm sure the students have gotten what you have said and your experience for Med 2. Thank you very much, and have a good night.
Thank you very much, and I wish you all the best, guys.
Good evening, Doc.
Good evening, Doc. How are you?
I'm good, thank you. My apologies. I had the power cut, so I couldn't make it to class on time. I'm so sorry.
Okay, it's fine. Actually,
Yes.
you can start talking now to the students. So, um, students, this is Davis, and is is a med three medical student, and is actually your senior, and is going to speak to you just like Annie did, and is going to share with you his experience with Med 2. He's actually my student. He was my student, and he's still my student. So, Doc, you can go
Okay.
on.
All right. Thank you so much. Good evening, everyone. I'm hoping I'm clear enough, and everyone is able to get me loud and clear. Am I clear enough? Can someone just confirm that I'm audible enough?
Yes, you are.
Yes,
Okay.
you
Yes, you
are.
are.
So my name is Davis Mkosha, a third year medical student. So before I even go any further, I would like to congratulate you guys for making it this far. It's not easy, and you should be proud of yourself because it's not easy. If it was easy, everyone was going to be here. So congratulations, guys! You've come a long way, and I want you to continue working hard and putting your very best. So yeah, going direct into what I would like to talk about, I will not waste much of your time as my colleague Annie had highlighted most of the things. I just want to maybe talk about a few things, maybe two or three things. Yes. So, honestly speaking, guys, it's second year is really challenging. If you're coming with a mentality believing that it's going to be as easy as it was at first year, I want to advise you to change that mentality. And trust me, you really need people to guide you. Why am I saying so? Well, personally, from my experience, what I saw was I really had a challenge with managing my studies because there's a lot of work to do, guys. There's a lot of work to do. You have about 7 courses, and in all those courses, they will need your equal attention, guys. Trust me, the moment you begin to neglect certain courses and you're giving much of your attention to other courses, that becomes a very big problem, and you might have a challenge. You might have a challenge, and that will be very bad for you. Now, before you even realize that you're failing or you're having a challenge in 1 or 2 courses, there'll be a lot of assessments coming in. As a result, you might ruin your CA, or maybe you might find yourself in a situation where things will be very bad and it'll be difficult to make it in the exam. Therefore, I would like to encourage you guys to really be part of the SA Academy Tuition Center because personally my experience was it was easy for me to go about certain things because I was guided accordingly by someone who has been there and who knows how these things come. So in as much as I had a lot of work to do, a lot of things to study, but at least from the emphasis I got from Dr. Shadrick, who helped me in not only biochemistry, physiology, and some anatomy as well. And by the way, I cleared all my courses, and I'm glad to mention it. Of course, I know others, my fellow, the people I was with last year, they did not make it, which is very unfortunate. But trust me, the competition is there, and you really need to be hardworking, guys. You really need to be hardworking. And trust me, had it not been for the help I got from Dr. Shadrick, I don't think, honestly, I would have been talking to you right now. Yeah, so this is why, guys, it's very important that you remain dedicated to your books. You should know what you're supposed to do. Then, most importantly, guys, be consistent, guys. Be consistent. I don't know. Am I clear enough, guys? Can you hear me?
Yes,
Yes,
you
we
are
can.
clear.
Yeah, yes. Okay, thank you so much.
You're
So
clear.
yeah, yes, guys. So be consistent, guys. There is nothing difficult. It is possible. People have made it, regardless of how many people have failed. I think you've seen, guys, up from like 1,167 people, only about 265 guys made it to, to third year. That's a crazy figure. It's very sad, but well, that's medicine. You really need to be hardworking. You really need to be hardworking, guys. Extremely hardworking. So now, already what you're doing, guys, belonging to SA Academy is already a step towards you making it to third year. I want to assure you this because I have been a product of Dr. Shadrick. He assisted me so much when I needed him. When I needed him the most, I remember one time he was telling me about because initially what he does is after his class he'll ask you some questions based on what you guys have covered. I always got bothered with that. I thought maybe it was a way of just bothering me instead of us just concluding the topic. Then it ends there. But trust me, all that is going to help you and it's going to carry you to your third year. It's important, guys, that you find yourself an academy, a tuition center. It's very important. You need guidance. You need someone to help you. These guys have been there. They've done this before. The mistake people make, guys, is coming into med school and thinking, believing that you do things by your own and you know how to go about everything. Well, of course, there are people that manage to do things by their own, but trust me, guys, it's extremely difficult. It's not even about how smart you are or maybe how much you do things by your own. You just need someone to guide you. Regardless of whatever program you might be doing, you need someone to guide you, guys. And this guidance that I'm emphasizing on, this is a guidance I got from Dr. Shadrick. He helped me so much, such that I could not, there are times where you're preparing for a test and you're very behind in your certain topics and you have to cover those topics at least before that test. So because of the help I got from him, he could guide me to say, this is what they'll ask you. You need to know this. This is a must. You need to know this. You need to know this. It was very easy for me to just get into my notes and go directly to the stuff that I was supposed to do because I know this is a must. This is what they will examine me on. So, these are things people do not know when they come into medical school. Things are bulk. Things are really bulk, and you might spend yourself maybe reading a lot of textbooks, unnecessary data, unnecessary information, but you're jobbing, guys, you're jobbing unnecessary data. But if you have someone to guide you, you'll find yourself studying the right data because you have someone who's guiding you, a senior who was once there, you see? So it's very cardinal, guys, that you belong to this academy. I really want to see each and every one of you making it to third year and just rejoicing by the time the year ends, Yeah, then that takes me to my last point, guys. Let me not waste much of your time. That takes me to my last point, which is knowing the right material to use, guys. Know the right material to use. Have the right material. I'm sure Dr. Shadrick has probably spoken about, if not, maybe he will definitely talk about what notes you're supposed to be using. Even the same notes that he gives you. Very important notes, guys. Know the material you're using. Do not just study because you feel this is the content that you're looking for, guys. Have the right material. Just, of course, study hard, but again, study smartly, guys. Be smart when you're studying. At least have some direction. This is why you need someone to guide you. Have a senior. It takes me back to being guided accordingly. If you've understood from the beginning of my advice, it's all taken me back to being guided accordingly. Yes. So guys, I'm wishing you the very best. I know it's not going to be easy. Trust me, it's definitely not going to be easy, but it is very possible. It's possible that each and everyone can make it, provided that you guys are hardworking, you're consistent. Most importantly, be consistent, guys. Do not pack your books and go, you forget. Be consistent, guys. Be hardworking. And yeah, you face a lot of challenges, but trust me, you can overcome all those challenges and make it to third year. Without a single doubt, do not be shaken by the numbers of people that have failed from my intake. You can make it. Do not be demoralized. What you're doing right now, this step that you've made to be part of SA Academy is already a step towards you going to third year. I can assure you this. It's very possible. I cleared all my 7 courses, and I'm believing you guys can make it as well. Thank you so much. I don't know if there are any questions, guys.
Is there anyone with a question to ask him before he goes? Yes, please. You can. Gordon, you can kindly unmute yourself and speak.
Thank you very much, sir. Doc, I just want you to tell us the strategy, the studying strategy. I can call them like that, like how you used to manage your time
My time.
and all
Okay.
the stuff. Yeah, thanks.
Okay. All right. Thank you so much. Deliberately, I didn't want to dig into how I managed to do and all that because I noticed my colleague Annie, I'm sure she talked much on that. But for the sake of going through that, let me just quickly mention it or highlight it rather. So, well, for me, like I mentioned at the beginning, I really had the challenge with managing my time, like having time for all the courses. Because these courses are really bulky. These are bulky courses and they need your equal attention. Like you can't forget about the other course and you're focusing on one thing. So, well, personally for me, number one thing was having a timetable, because I needed to give each and every course my equal attention. Having a timetable where I know to say, at least if I've studied this tomorrow, at least I should be doing this. If I've done this on such a day, I'm doing this. Then besides that, what kept me on track is the tuitions. I started tuitions very early because I needed someone to guide me. In as much as I am studying, at least I needed someone to guide me with the stuff that I'm studying. At least I need someone to emphasize to say, you need to focus much on these questions can come like this and this and this. Sometimes you can be reading your own notes, but you don't know how questions can be brought from those notes. So my main goal, I wanted to understand how questions were brought. In as much as I am about to study a certain topic, let's say for instance, maybe Dr. Shadrach is about to introduce a certain topic to you. Even before I study it myself, like personally, of course, I'll have an overview on it. I'll know it, but how are questions brought from from that topic? Dr. Shadrach would come in and assist me to say, these are examinable things. You need to know this, doc. You need to understand this. So yeah, having the right people around me, like Dr. Shadrach himself, like being there and being able to guide me and emphasize to say, these are things they'll normally ask you, know this, know how to go about this. Then besides that, one thing I forgot to mention, my apologies for that, for that, having a reliable study group, guys. Study groups really help. If you never had a study group at first year, please make sure that you belong to one. Do not be too many. Personally, our study group was just the 3 of us, and we really found it to be very productive. But I don't know, it's up to you. People have different preferences. Others would say, no, us want to be 5. At the end of the day, it depends with what works for you. I might tell you we're the 3 of us, but then you might find not that to be working for you. But that's what worked for myself and my study partners. And from the study group I was in, all of us cleared. No one has a supplementary exam. No one is repeating it. All of us cleared, guys. Yeah. So belonging to a study group really helps. It really helps. And yeah, I'm not so sure if I've answered your question. I hope I'm not talking too much and I haven't answered your question.
You have, sir. You have, Doc. You have.
Thank you so much, Dr. Good. Thank you so much.
All right. Thanks, sir.
Yeah. Do we still have some more questions, guys? I think I can spend 1 or 2 minutes before Dr. Shadrach begins teaching.
Do we have anyone with a question before he goes? Okay, I think there is no one with a question. Doc, thank you very much for that advice to the students, and they'll live to remember.
Thank you so much, sir.
Otherwise, we are releasing you.
Thank you so much, sir. Thank you so much.
All right. Have a good night.
All right, good to have you.
Okay. Good evening once more. So for those of you that doesn't know me, I'm Shadrach, and I'm a 7th-year medical student from Ridgeway. I've been teaching medical biochemistry and genetics for too long now. So I kind of appear to be more familiar with the questions that the examiners would actually ask you in this course. Okay, so welcome to SA Academy, and you are actually free to consult any one of us with any problem that you are going to face throughout the academic year. So to introduce you to 2nd year, I'm first going to congratulate you for making it to 2nd year, because it was never easy. People failed, and we see people writing supplementary exams. Some are repeating the year, but you guys have managed to cross over to 2nd year without any much problems. So your journey of medicine begins now, and it begins from today. So you should tighten your belts, because medicine is not natural science. So the courses that you are taking at first year, they are kind of natural science, not really medicine, but natural science. This time around, these are the first medical courses that you are taking. Therefore, 2nd year and 3rd year, we refer to them as basic sciences, because they are going to help you in the application of understanding diseases on the wards. Okay, so it's very important for us to pay attention to detail. Okay, pay attention to detail, and also you should know exactly what you are studying. You should be oriented to time, place, and person. Okay, so the first course that we are going to deliver to you is medical biochemistry and genetics, for obvious reasons. One, many people didn't know that actually this course was a killer course. Students thought that it was an half course, so they were not even taking it seriously. Okay, so many of the people that didn't pass this course, it's because they thought it was a half course. Okay, I met a student who was talking about medical biochemistry being so boring, and so they couldn't study it until the final exams showed up. Actually, they used to give more respect and time to immunology and microbiology, which is totally the opposite, because as you can see, PGY2320 is a full course. So any course that ends with a 0 at the end, just know that it's a full course. So 2nd year has got 7 courses. The 7th course is a project course. Out of the 7, we have 6 examinable courses. But even the 7th course, the DME course, is also examinable, but it doesn't have points. Okay, only 6 courses out of the 7, they have points. From out of the 6, you have 3 which are full courses, and the other 3 set which are half courses. So PGY 2320 and PGY 2310, HAN 2210, these are full courses. PTM 2515 is an half course. CHE 2501 is an half course, and of course PSY 2415 is an half course. So this one is a full course, and it has to run from January all the way to December. Okay, because it's a full course. Right. So this one has a lot of casualties, guys, from the 2021-2022 academic year. It has so many casualties, and that's the reason why we are starting with PGY 2320. Now, PGY 2320 is not chemistry, guys. It's a physiology. So
it's from the physiology department. Okay, so that's the reason why you see the course code is PGY and not CHE, because this is not chemistry, this is physiology. Okay, so medical biochemistry and genetics has two components. The first component is medical biochemistry, the second component is genetics. Right. Now, the medical biochemistry part has four sub-components inside it. And in order, we have structural biochemistry, we have enzymology, we have biochemical, we have metabolic biochemistry, and of course, the last one should be medical genetics. Okay, so the medical biochemistry component has three sub-components. I'm repeating: structural biochemistry, enzymology, and metabolic biochemistry. Then the genetics part, it has other small minor topics inside. So for us, we are starting with medical biochemistry. The genetics component will be towards at the end of the academic year. So for now, test 1 and test 2, they'll be focused on medical biochemistry. In the group, I sent a list of topics that comes under each test. Test 1 will be your structural biochemistry plus enzymology. Test 2 will be your metabolic biochemistry. Test 3 will be medical genetics. Right. So you guys, you are oriented. So the topic that we're about to do today, the first topic is coming from structural biochemistry. Here we'll be dealing with structures. It will be a combination of Bio 1400 and CHE 1010. So we are combining the principles of chemistry to understand how the cells are able to function. Right. So whatever we're going to learn from today until the time we're going to start doing enzyme structure and catalysis, it will be structural biochemistry. Okay, it will be structural biochemistry. So the first topic in structural biochemistry should be biophysical chemistry for obvious reasons. Okay, so biophysical chemistry, when you open the details of this class, you'll see what biophysical chemistry is. It's a physical science that applies the principles of chemistry to understand how the cells are able to survive. Okay, it's a physical science that enables it's a physical science that uses the principles of chemistry in the understanding of how living cells survive. Okay. So this is a very important course for obvious reasons. If you are going to understand biochemistry, it will be very easy for you to understand pharmacology and therapeutics next year. For you to understand how a drug works, you need to understand the biochemistry behind every certain disease process. So any drug that you are going to give the patient, it's obviously going somewhere, and there's obviously there are obvious things happening in the cells. Okay, so here you're going to learn how the cells are able to survive with regards to the pathways, the enzymes. Okay, so, and then next year you'll be taught about how the drugs works and everything else. So you need to understand this for you to understand what's going to happen next academic year. Okay. So biophysical chemistry, once more, is a physical science that uses the principles of chemistry to understand how cells survive. So in this topic, we are going to look at how the structure of water and also the properties of water. We're also going to look at pH and its calculations, and last but not the least, we'll look at the buffer system. So by the end of this topic, students should be able to explain the following concepts. The first one is that you should be able to tell us the properties of water as it being a solvent for biochemical reactions. So a chemical reaction happening in a living cell is called a biochemical reaction. So the bio part is the living part, then the chemical part is the chemistry. Okay, so biochemistry is not organic chemistry. Biochemistry is this the use of chemical principles to understand how cells survive. Okay, so we have so many pathways in the body, so many pathways taking place, even as at now. We have so many things like the glycolytic pathway that breaks down glucose to produce energy in form of ATP. We have amino acid metabolism. We have so many things happening in the body at once. Okay, so it's very important for us to understand um, the properties of water, because this is where the chemical reactions, the biochemical reactions takes place. So we have to understand the properties of water as a solvent for biochemical reactions. We also have to understand hydrogen bonding, because this water molecule is going to be interacting with other polar compounds like amino acids, okay, and even non-polar compounds. We are also going to understand the weak acids and also the weak bases, which is of course going to help us to understand the buffering system, because cells they do necessarily they don't survive if the pH is not optimum. Therefore, we shall also look at the pH and also the pKa determinations, and also we are going to look at the buffering system, so the buffers and their role in biological systems. Last but not the least, we're going to look at the physiological important buffer system, like the bicarbonate system. Right. Okay, so we can chip in. Gordon, you have a question? I can see your hand is raised.
Oh, no, sir, it's just a mistake.
Okay. So our journey begins now. So this looks like it's Bio 1400, guys, but it's not. Trust me. So it looks like Bio 1400 because Bio 1400 is a prerequisite to this course together with CHE 1010. So they will kind of like look like it's Bio 1400 and CHE 1010, but it's not. Okay. So the first thing that we need to understand is water and its structure and the properties. You guys are just fresh from Bio 1400. So we know that water is the most abundant substance on Earth. So as you can see, this picture here shows an ocean. Okay, you can see that almost 70% of the Earth is water. So there is more water than land. Okay, so so water is the most abundant substance in the living systems, which is the human being in this case. So tomorrow, Tutor Frank is going to start human anatomy and development. So you guys, you start learning about the human being. So I, for one, I don't teach anatomy. I think Davis just mixed up things. So so water is the most abundant substance, okay, in the living systems, which makes up to 70% or more of the weight of most organisms. Again, in PGY2320, I'm going to teach you about the body fluid compartments on Wednesday. So, guys, you should understand that 70% of your body is water, okay? Obviously, the first living organisms, they actually arose from the aqueous environment, from water. So we're actually coming from water, okay? So all aspects of the cell structure. So the cell structure and its function, they're adapted to water as a major solvent, okay? And also the attractive forces between the water molecules and the slight tendency of the water to ionize, because we know that water has got that tendency to ionize, okay? A crucial important thing to understand, okay, to the structure and also to the function of biomolecules. Remember, in Bio 1400, we learned about the carbohydrates, we learned about the lipids, the proteins. Okay, so biochem is actually, it starts like bio 1400 because we have biomolecules. What I called as structural biochemistry is actually biomolecules in 1400. So the water molecule and its ionization products, which are the protons and the hydroxide ions, and also the non-covalent interactions, the hydrogen bonds, are responsible for the strength and also the specificity of the biomolecule. So the biomolecules, they depend on the hydrogen bonds, they depend on the non-covalent interactions, they depend on the ionization of water into the protons and also the hydroxide ions. So the solvent properties of water, which includes its ability to form the hydrogen bonds, is actually very important. Okay, so water is actually able to form hydrogen bonds with itself and also with other solutes, with other biomolecules. Okay, and that is very important because most of these biochemical reactions, they are happening in water. So we talked about the weak interactions with regards to water molecules, which we know we have hydrogen bonds. We emphasized much intensity about the hydrogen bonds, guys. So we should also remember that the hydrogen bonds, they occur between water molecules, and they actually provide the cohesive forces that makes water a liquid at room temperature and pressure. And this favors the extreme ordering of the biomolecules. Okay, and that is actually very critical of the crystalline water molecule, which is ice. Okay, so hydrogen bond, they are very important when you want to consider ice as a part of the state of the water molecule. Then the polar biomolecules, they actually dissolve readily in water, and because of this, they can replace the water-to-water interaction. So the biomolecules, they can actually break the water-to-water interactions, which are more energetically favorable water-to-solute interactions. Okay, because we know that the proteins, for example, the amino acids will actually be the bond between the amino acids and water will actually be more stronger than the water-to-water interaction. In contrast, the non-polar biomolecules, they actually interfere with the water-to-water interactions, but they are able to form the interaction between water and that particular solute. And consequently, the non-polar molecules, they're actually poorly soluble in water. Okay, so we know of so many biomolecules that are insoluble in water, for example, the lipids. Okay, so in aqueous solutions, the non-polar molecules, they tend to cluster together. For example, the lipids, they will cluster together and they form the droplets. So this table, you can see that we have the melting point, the boiling point, and also the heat of vaporization of some common solvents. You can see that water, being a small molecule, it actually has a way large melting point and also boiling point as compared to other molecules which are non-polar. Okay, you can see that even the heat of vaporization is actually way bigger than the other compounds. Okay, so before we move on to the properties of water, we need to understand its structure first. Okay, so this course looks like it's Bio 1400. Someone can actually be tempted to think, oh, this is Bio 1400, so I don't have to study this topic. But hey, don't do that. Okay, don't do that, because this is the basis for understanding the metabolic chemistry, which we'll start dealing with, I'm sure, some 7 weeks from now. Okay, so water is a molecule that is made up of an oxygen atom with other two hydrogen atoms. You can see on this beautiful diagram, this blue thing, which is the oxygen atom, is chemically combined with two hydrogen atoms at a certain angle. So there's this angle which is created, and you guys, you know, it is 104.9. So one molecule of water is actually made up of two hydrogen atoms that are bonded with one oxygen atom. And we know the molecular formula of water is just H2O from basic chemistry. Okay, you can on this diagram you can see the water molecule, and you can see the electrons in the various energy levels, and this is just the same as this diagram that you saw at first. Okay, so the bond that forms the water is actually a covalent bond. So that bond that you are seeing between the oxygen and the hydrogen is actually a covalent bond. So we need to know that water is a polar molecule and it has oppositely charged ends. Okay, so one end is positive, okay, partially positive, which is the hydrogen, and this end here is partially negative, and this end here is partially positive. Okay, so we already know that water consists of oxygen and two hydrogen, and this is just to show you the Bohr model for the water molecule. So you can see from this diagram the electrons that are spinning, and also the partial positive and the partial negative on the oxygen. Okay, so the hydrogen has got it doesn't have any unpaired electrons, and it actually shares all its electrons with the oxygen, and it leaves the molecule with the positively and also the negatively charged ends. This is very important when it comes to the interactions with other biomolecules. Okay, so the bond angle, as you can see here, it's 104.5 degrees, which is very important because it gives water a specific shape which allows it to interact with other molecules. Okay, so very important to remember the bond angle and also the distance between the two hydrogen atoms. So the distance from here to the distance from here to here is very important. That is the hydrogen bond, which is 0.11 nanometer, and then the distance from here to here, that's the covalent bond, and this is equivalent to 0.0965 nanometers. Okay, so an electrostatic attraction between the oxygen and the hydrogens that you have seen is actually a hydrogen bond. Okay, so the hydrogen bond is not this one here. This one is a covalent bond. The hydrogen bond is this one between the hydrogen and the oxygen. That's the hydrogen bond. Okay, so the hydrogen bonds are actually relatively weak as compared to the covalent bonds. So those in the liquid water, they have a bond dissociation energy, okay, which is actually the energy you need to break that bond. Okay, and it's about 23 kilojoules per mole as compared to the 470 kilojoules per mole for the covalent oxygen to hydrogen bond. So the covalent bond is much more it's very difficult to break the covalent bond than it is to break the hydrogen bond. Okay, so you should also know that the tetrahedral arrangement of the orbitals about the oxygen atom actually allows each water molecule to form the hydrogen bonds with as many as 4 neighboring water molecules. So this exists in a crystalline state, like in ice. You are going to have 4 hydrogen bonds per water molecule that you have. But in liquid form and at room temperature and pressure, the maximum amount of hydrogen bonds is just 3.4. Okay, so this is the tetrahedral arrangement in ice. Okay, when water exists as ice in the crystalline form. Okay, so each water molecule is allowed to have a maximum of 4 hydrogen bonds: 1, 2, 3, and 4. Okay, so that's just the same thing. Okay, and you should also remember that water actually forms the hydrogen bonds with polar solutes, as in biomolecules, like amino acids. So they actually readily form between an electronegative atom and a hydrogen atom, which is covalently bonded to another electronegative atom. So as you can see, this is a water molecule down here, okay? And the water molecule is donating the hydrogen. So we have the hydrogen donor and we have the hydrogen acceptor. So molecules like ketones, nitriles, um, carboxylic acids, amino acids. As you can see, these molecules, they actually have hydrogen bonds with water, okay, which is very important. So the hydrogen bond can form between oxygen and hydrogen, nitrogen and hydrogen, oxygen and hydrogen, oxygen and hydrogen, and oxygen and hydrogen, and as well as nitrogen and hydrogen. So it's very important to remember the hydrogen bonds because they're the ones that brings about so many properties that water has. Okay. So the hydrogen atoms, we know they are covalently bonded to the carbon atoms. Those which are covalently bonded to the carbon atoms, they actually don't participate in hydrogen bonding. Why? Because carbon is only slightly more electronegative than hydrogen, and therefore the CH bond is only very weakly polar. It's not even polar, it's actually non-polar. Okay? So the difference will actually be explained. Will actually explain why butano. This is butano. It's an alcohol. It has a relatively high boiling point of 117 degrees Celsius, whereas butane, which doesn't have the oxygen, the boiling point is only 0.5 degrees Celsius. So it's very important to remember the hydrogen bonds. Okay. So alcohols, aldehydes, ketones, and also compounds that contains the nitrogen-hydrogen bonds, they will all form the hydrogen bonds with water molecules, and they will actually tend to be soluble in water. So these are the things that you are going to learn in organic chemistry: alcohols, aldehydes, ketones, and various other functional groups. Okay, so here you can see this is water and this is an aldehyde molecule. This is an alcohol. This is water bonding with a ketone. So this is a ketone, and this is an amide. Okay, so hydrogen bonds, we know they form between oxygen and hydrogen. Okay, so we also see between the complementary bases of DNA, which will be learned under medical genetics, you can see that thiamine actually bonds with adenine, A to T bond via the hydrogen bonds. You can see the hydrogen bonds like that. Okay, before I continue, any questions? You can just unmute yourself and speak if you have a question.
All right. Thank you very much for that presentation. It's awesome. My question is just goes to the first part where you were talking of aldehydes and all that form of structures. Are we supposed to know how to draw those structures, or we should just know the bonds that are involved?
Okay, it's a very good question. So here you're not restricted to anything. So this is structural biochemistry. So you should know obviously how to draw things. Okay, this is structural biochemistry. So you should know how to draw these structures. So yes, you should know how to draw the structures.
Thank
Is
you.
that clear?
Yes.
All right. So Gordon, you have a question? I can see your hand is up.
Oh no, sir. I can't just move the hand, but I'm not. I haven't raised any hand. I don't know how to like move it from the time I raised the
Oh,
hand. Yes.
okay. Well, okay. So what actually readily dissolve most biomolecules, just like we said, like proteins, carbohydrates, they'll be able to dissolve in water. Okay. And generally it's actually charged. Okay, it's actually charged, and it will actually bond with other molecules that are also charged. So the compounds that dissolve easily in water, we are going to give them a name. They should be called hydrophilic. Anything that dissolves in water should be called hydrophilic. Okay, and anything that does not dissolve in water should be called hydrophobic. So hydrophilic molecules, they are going to dissolve in water. But hydrophobic molecules, they are not going to dissolve in water. For example, lipids and waxes, they don't dissolve in water. So the water will actually dissolve salts such as the sodium chloride by hydration and also stabilizing the sodium and the chloride ion. So there will be hydration and stabilization, which weakens the electrostatic interactions between the sodium and the chloride, and therefore counteracting their tendency to associate with a crystalline lattice. We don't want that kind of arrangement in the sodium chloride. So there'll be hydration, then stabilization. The same factors will also apply to charged biomolecules such as carboxylic acids and protonated amines. Okay, they will also do the hydration, they'll be hydrated then stabilized with regards to water. Okay, so this diagram just shows you the interaction of water with charged solutes. So water is going to interact, for example, with sodium chloride, hydrate it, then stabilize it. So there is hydration, then stabilization. Okay, so we continue. Now, this you need to remember, and you need to tell us the reasons why you are saying that water is a polar molecule. Why? Why is there cohesion and why is there adhesion? Why is there surface tension? Why is there capillary action? Okay, so this is very important. Why? Because these biochemical reactions, when they're happening, they're actually producing heat. If that heat is going to continue, it may actually kill the cells. Okay, it may actually kill the cells. If the temperature goes up, the cells can actually die from hyperthermic dehydration. So on the properties of water, we need to know, when you mention a property, you need to tell us why it exists. And most of these properties of water, they can be explained by using the hydrogen bonds. Okay, so now this is where I pause and ask the students to tell us the properties of water one by one, and then explain why that property exists. So this is where I stop. Even last year, I stopped here when teaching Bennett Davis. So I stopped here and asked the students to tell us what are the properties of water. So I've stopped. I need volunteers to start speaking. So you can just unmute yourself. You say a property of water, then you tell us why it exists. Okay? Any volunteers? Blessing, are you in class? Blessing, unmute yourself.
Yes, sir.
Yeah, Doc. No.
Yes, sir.
Blessing is there. Yes, Blessing, yes. Tell us one property of water that you remember from Bio 1400.
Water is a universal solvent.
Okay, so it's a universal solvent,
Yeah,
meaning
which
what?
means it allows substances to dissolve in it, and it also allows for a good transport medium.
Okay, that's very good. And what allows it to dissolve most of the substances?
The hydrogen bonds.
Yes, it's the hydrogen bonds, as you have seen in the previous slides. Thank you very much, Blessing. Next volunteer, please.
Yeah, this is Mukata.
Okay,
Yes,
Mkata,
Doc.
yes.
For me, I pick one of the property of water is it has a cohesion and adhesion forces. So those two, they helps in the movement of water molecules, or rather any other fluids, like when the way the blood moves in the blood vessels. So those properties are the one that are responsible, because they attract to each other and attract to the walls of the blood vessels. So that property of water, the one that helps the blood to move in the blood uh, in the blood vessels. Yeah.
Okay, 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 Mkata. So he's mentioned something, cohesion and adhesion. I want someone to tell us what adhesion is and what cohesion is, and what's the difference between the two. Eliza, can you unmute yourself and tell us what cohesion is?
I think cohesion is the ability of water to attract with other water molecules. And
Okay,
then
and
adhesion is the ability of water to attract um, with with other
sorry, come
with
on.
other molecules. I said
Okay, you're saying
adhesion
adhesion is,
is the
yes.
ability of water to attract with other molecules.
Okay, someone to put it in a better way. Adhesion, cohesion was correct.
All right. Can I go on?
Yes, go on, please.
For me, I can. The way she has said it, but I'll just add on is cohesion. It's a bit where substances of similar molecules uh, converge or attract each other. While for adhesion, it's where a substance of different molecules cohere or attract each other.
Sorry, come again with adhesion.
I said adhesion is where the ability in which substances of different molecules attract each other.
And cohesion?
For cohesion, it's the ability of substances of similar molecules attract each other.
You're kind of confusing us. Uh, I saw some hands. You can kindly unmute yourselves, please.
Yes, sir. I'll explain surface tension, but first of all, I would love to talk about the cohesion and adhesion thing. So surface tension, I
Yes.
think, is brought about due to cohesion, because cohesion is the ability of water molecules to bond to other water molecules. Let me say like that. Let me give an example. If you have water in a cup, and then on let me say on some a cold day, you might see maybe insects working on water and stuff. That is due to surface tension, and that surface tension is caused because water is able to is able to make some cohesion forces with other water molecules down there instead of the air particles that are on the layer of the water. Let me say, because if you have, let me say, water in a cup, they have made the layer like there's air above the water and there's water down the cup, like in the cup. So there's tension. The water is only able to bond with the other water in that cup, but it can't bond to the air like above the water level. So that's what brings about the cohesion. That's what brings about surface tension as a result of cohesion. I think maybe I'm clear with.
Yes, so that's exactly what we wanted. So cohesion is where we have like molecules attracting to each other. Adhesion is unlike molecules, like water with the surface. Okay, so both of these, they are going to help in surface tension, to create the surface tension. Okay, very good. Next property of water, please. Anyone who is available to speak, you can just unmute yourself.
Okay, good evening, everyone.
Good evening to you too.
Yes, though Charlie about, I would love to talk about capillarity.
Okay.
Yes, so with capillarity, this is a property of water whereby water molecules are able to flow or pass through very tiny or narrow, if I can say, narrow tubes, or because of lack of better terms, yeah.
Okay. And what brings about the capillary action?
Okay, so I believe it is the same cohesion and adhesion properties, if not mistaken.
Yes, so a combination of adhesion and cohesion, they will give us the capillary action. Any other properties of water that you remember? Okay, so water has actually, it has a very high specific heat capacity as compared to other substances. So the amount of heat energy required to vaporize water is actually very, very big as compared to other molecules. Okay, so you can see here, water actually has the greatest density is at 4 degrees Celsius. Okay, so the greatest density of water, you have to remember that it's at 4 degrees Celsius. And Bressings talked about it being a universal solvent of life. Someone mentioned the capillary action, which is due to both cohesion and adhesion. And someone mentioned about surface tension. So there's also bions. For example, the CSF fluid, which is found in the brain, actually keeps the brain floating in some kind of a fluid, which is water. Okay, so the bions is very important, especially in the central nervous system, because it keeps the brain bouncing without any injury happening to the brain. Okay, so water is also a polar molecule, which is a known thing. We mentioned adhesion and cohesion, and I mentioned the high specific heat capacity of water. Okay, so each of those things must be explained. Guys, in the exam, they are going to ask you, um, uh, mention two properties of water and explain how each of those properties comes about. Or they will mention the property of water and then they'll ask you to explain how it comes about. So the exam actually starts from right here. Okay. So we need to remember what's going on starting from there. Now,
All
the
right,
other
sir.
thing that you need to know is that water undergoes. Yes, Mike.
Uh, yes, I really wanted to ask as with regards to that property which saying water has its greatest density at 4 degrees. I mean, what really happens? Why does it have the greatest density at 4 degrees?
Sorry, come again.
Um, there's this property saying water has its greatest density at 4 degrees.
Yes.
Yeah, so what really, why is it that it has it's what greatest density at 4 degrees? What really happens? I don't know if it's maybe the molecules or something.
No, nothing. No, nothing happens. It's like the boiling point of water being 100 degrees. It has no explanation. Nothing. It's just the thermometer. You get the thermometer. You measure the temperature, and you're actually also measuring the density. There's no explanation whatsoever. You can't explain why the the, the boiling point of water is 100 degrees, for example.
All right. All right. I
I hope you.
understand. I get.
Yes. Yes, there was another hand. Someone wanted to ask, what is bryons? Oh, you guys actually knew from physics.
Um,
Um,
I just wanted to
you
ask
guys,
something.
yes.
Uh, can you also include, like, in
Go on.
terms if you're asked to mention any properties of water, can you also? I was looking at the list, the properties that you mentioned, and can you also include it has a high heat of vaporization? Is it
Yes,
also
I
okay?
actually mentioned that water has high heat of vaporization.
All right.
I did mention that, and that is true. Have I answered your question?
Yes.
Okay, so someone is asking what is biomes, because obviously biomes is a property of water. So anyone, because you guys are new from physics, just fresh from physics, anyone who can help us with what biomes is? If you remember, just unmute yourself.
What about
Okay,
something? Hello.
sorry.
Yeah. What of the ability of something to float in water?
Yes, exactly. So the ability of a substance or object to float in a fluid, actually it's not just water, it's fluid. So it could be anything. So that's biomes. So for example, the brain is able to float. The brain and the spinal cord, they're actually floating in water. That is biomes, and it's very important because it protects the brain anyway from injury. I hope I've answered your question. I hope we have answered your question. All right, Miracle. You're welcome. Okay, so we should also understand that water can undergo ionization. So ionization just means the ability of a substance to undergo um, to undergo a change which leads to the formation of ions. In this case, we have the hydrogen ion being formed and the hydroxide ions. So as you can see here, water can actually has a slight tendency to undergo reversible ionization. You can see this is a water molecule. There's a forward reaction and a backward reaction, which leads to the formation of hydrogen and the hydroxide ions. Okay, so ionization is very important, very, very important because it produces the hydrogen ion, which is a proton, and a hydroxide ion, which gives us the equilibrium equation. So you can see that equilibrium equation on the previous slide. So although we commonly show the dissociation product of water as the H plus the free protons, actually they do not exist in solution. Why? Because they'll be combined with other molecules. So the hydrogen ions formed in water are actually immediately hydrated to hydronium ion, which is the H3O plus. Okay, so the H plus does not exist because the hydrogen undergoes hydration to form the hydronium ion. Okay, so hydrogen bonding, which occurs between the water molecules, it actually makes the hydration of dissociating protons instantaneous. So immediately, it's fast. It's a fast thing. Okay, so as you can see, two water molecules here with the hydrogen bonding there. You can see the hydronium ion and the hydroxide ion. Okay, and this can continue making these structures you can see here. So it's very important to understand that ionization will take place and it will lead to the formation of hydronium ion. So we can measure ionization of water by electrical conductivity. So we know that pure water, for example, it carries electrical current as the protons, they migrate towards the cathode and also the hydroxide towards the anode during electrolysis. So the movement of the hydronium and the hydroxide ions in the electric field is actually anonymously fast as compared with that of other ions such as the sodium, potassium, and chloride. So we actually don't have protons that moves very far through the bulky solution, but we have a series of protons that actually whoops between the hydrogen bonded water molecules. And this causes a net movement of a proton over a long distance in a remarkably short time. So we do understand that the hydrogen that is formed as a result of ionization actually will cause a net movement of the protons over a long distance in a short period of time because of the ionization. So as a result of this, the high ionic mobility, okay, the acid-base reactions in aqueous solutions, they are generally exceptionally very fast. So those reactions that happens in water associated with the acid-base reactions, they are extremely fast, okay? The reversible ionization is actually very crucial to the role of water in cellular function. So it's very important that water actually undergoes reversible ionization with regards to how the cell operates. Okay, we can actually quantify the ionization of water. As you can see, the position of equilibrium of any chemical reaction is given by its equilibrium constant, which is KEQ. So for example, we have a reaction where A is reacting with B to produce C and D. We can formulate an equilibrium constant equation. So this is an equation where we have the product of the multiplication of the product over the multiplication of the concentrations of the reactants. Okay. In this case, morality is the unit of concentrations which is used to calculate the equilibrium constant. So this whole equation will give us the equilibrium constant. In the same regard, we know that water undergoes ionization. We can actually form an equation which is the equilibrium constant equation, where we have the product of the concentrations of the products, which is the hydrogen and the hydroxide, over the concentration of water. Okay, but you must remember that pure water at room temperature and pressure, the concentration is actually 55.5 molar. That is for water. Okay, so when we rearrange the equation, we realize that here we're going to put this over 55.5, and the value of the equilibrium constant will be determined by electrical conductivity. So some experiments were done, and it was found that the equilibrium constant was 1.8 by 10 to the power minus 16. That is at room temperature and pressure. When you substitute this value for equilibrium, just a minute. Okay, just a minute. I log out on this device. Okay, so we'll see where the equation is going, because it's a very important thing to know. It's very much examinable, and you guys are going to submit an assignment which will be given to you at the end of the class. So you're going to see that Just from there, we're going to come up with so many things. Just from that equation, we're going to come up with so many things. Okay. Okay, so we already saw this. So when you rearrange this equation, we have been told that the equilibrium constant for water is 1.8 times 10 to the power minus 16 molar. So when you multiply that with 55.5, it will give you 1.0 times 10 to the power minus 14 square molar. Okay. Okay, the equation is this one. So the equation is this one. You start with this. This is coming from ionization. So the KQ is equal to the product of the proton times the hydroxide over 55.5 molar. So the 55.5 molar is actually, it was actually measured. It's a concentration of water at room temperature and pressure. Okay. And the KQ was calculated by electrical conductivity, and it was found to be 1.8 times 10 to the power minus 16 molar at room temperature and pressure. So when you put that value on Kw in this equation, when you put the 1.8, you are going to multiply with a 55.5, which will be equal to the product of H plus and hydroxide. So this whole thing will give us 1.0 times 10 to the power minus 14 molar squared. Okay, so take a look at this. Therefore, the product of the protons and the hydroxide ions in aqueous solutions at 25 degrees Celsius is always equal to 1.0 times 10 to the power minus 14 molar squared. So when there are exactly equal concentrations of the hydrogen and the hydroxide as in pure water, the solution is said to be at a neutral pH. So if you have the equal concentration of the hydrogen and the hydroxide, it means that that solution is at a neutral pH. Okay. For example, the pH of blood is slightly neutral, not exactly, but slightly. So at this point, the pH, the concentration of the hydrogen ions and the hydroxide ions can be calculated from the ion product of water as follows. So we can calculate the pH, and we can also calculate the concentration of the hydrogen and the hydroxide ions. So here you can see Kw now is equal to this. And if the concentration of the hydroxide is equal to the concentration of the hydrogen, it means that we can say Kw is equal to H plus squared. And when you solve for H plus concentration, it will actually give you this. So it will actually be square root of Kw. And Kw, we know that Kw was actually 1.0. So remember, it wasn't at first. It actually wasn't Kw. It was just something else. It was K equilibrium. Then it turned into Kw because the W is for water, the equilibrium constant of water. So square root of Kw, which is the square root of 1.0 times 10 to the power minus 14, is actually 10 to the power minus 14 molar. So this is the concentration, equal concentrations for H plus and also the hydroxide ions. So it's very important because we're going to use this same concentration to calculate the pH. So we're going to come back for the pH, you're going to see. So now sometimes you should actually remember that water is actually a reactant, can act as a reactant direct in these biochemical reactions. So again, it's very important to remember that you can actually add water to a compound, or you can remove water from a compound. When you add water to a compound, that particular compound will undergo hydrolysis. So that will be a hydrolytic reaction. Very important to remember. Why? Because we have enzymes that are actually helping hydrolytic reactions to take place. Okay. Again, you should also remember that water can be produced in these biochemical reactions. When a reaction is happening, a reaction pathway is happening, it can actually produce water. And that reaction is called a condensation reaction. So water is actually both the solvent in which metabolic reaction occurs, and it's actually a reactant in many biochemical reactions or biochemical processes. That includes hydrolysis, condensation, and also oxidation reduction reactions. So reactions such as hydrolysis, condensation, and also redox reactions, water can act as a reactant. So it can directly react with other things, or it can be produced from those particular reactions. So water is not just the solvent in which the chemical reactions of living cells occurs. It's actually very often a direct participant in those particular reactions. Okay. For example, the formation of ATP from ADP using the inorganic phosphate is an example of a condensation reaction. So a reaction where water is produced is actually a condensation reaction. A reaction which utilizes water is a hydrolytic reaction. In biochemistry, don't just mention things anyhow. When you see water is being produced from a reaction, you should say that that's a condensation reaction. You see a reaction using water, that's a hydrolytic reaction. So for example, the formation of ATP from ADP, you can see ADP, this one is a reversible reaction. So we are talking about the forward reaction starting from ADP. So from ADP, you can see that water is produced. Here, water is produced, meaning this reaction is a condensation reaction. But the forward reaction, you can see that the forward reaction for us here is this one on top. The forward reaction, water is being used and it's breaking apart the bonds, okay? So this one is a hydrolytic reaction. Right, so the reverse of this reaction, which is the cleavage accompanied by the addition of elements of water, is actually a hydrolytic reaction. So for this reaction, its forward reaction is down, then the reverse is this one. So what we are telling you is that the forward one is condensation, the reverse is hydrolysis. Okay? So the metabolic water, which is formed by oxidation of foods, for example, and also stored fats, is actually enough to allow some animals in very dry habitats, like in deserts, okay, to survive. So because of that water which is produced in those reactions. And also the green plants, they utilize water to produce energy. Okay. Right, that was a long journey talking about water and its properties and also the ionization. Now it's time for us to chip into the pH. This is where we're going to see how to calculate pH from the Henderson-Hasselbalch equation. A very, very important equation to remember, a very, very important equation, because you'll be asked what the Henderson-Hasselbalch equation is and actually state its parameters, and also how we are going to use the Henderson-Hasselbalch equation. Before we move on to pH, any questions on water? If you didn't understand anything, if you didn't understand something, of course, you should ask before we go on to pH. Anyone with a question?
So far so good. The presentation has gone well. I just want to ask on it is the previous part when we're looking at the hydrogen bonds, whereby you have for instance the compound that has the nitrogen in it, and we have water. How does it mean that 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 bond which exists between the water molecule and the compound that like a stock of nitrate, does it form hydrogen bonds, or which kind of bond which exists there between water molecule and nitrate thing?
Okay, the hydrogen bond will actually exist there because of the nitrogen, which is negatively charged, partially negatively charged, and the hydrogen, which is partially positively charged on the water molecule.
All right, thank you.
You're welcome. So we move on to pH. So the pH scale actually designates the hydrogen and also the hydroxide concentration. So the pH scale, it's actually a measure of the hydrogen and the hydroxide concentrations. So the ion product of water, which is Kw, is the basis for the pH scale. The understanding
hello.
of,
Sorry for disturbing.
yes, please.
Yeah, I just wanted to ask on the calculation part as well. Like, are you supposed to master this just like most of the time, like when you get used of it, you end up mastering, or maybe the way they ask question, they give you formulas. Like,
No,
yeah,
formulas are not given. No one gives formulas this time around. You have to master these things. Yes, so Miracle has said it. You have to know these things. This time around, there's nothing like you are going to be asked MCQs and you don't know things. No, you have to know what you are doing.
all right, thank you very much.
All right. So the pH, the term pH is actually defined by this expression. So that P is the potential, the H is the hydrogen. So hydrogen potential is equal to negative log 1 over H plus ions. Now, you know mathematics very well. It will give you minus log of H plus. So the symbol for P just means negative logarithm of, so this P is the negative logarithm of this. Okay, it could be P or H. Okay, it's not just pH, but POH also exists. So for a neutral solution, for example, we talked about this. The concentration of hydrogen should be 1.0 by 10 to the power minus 7 molar. And you can calculate the pH of such a molecule by doing minus log of 1.0 times 10 to the power minus 7, 7. And that would actually give you a pH of 7. So that's the reason why the pH of water, that's pure water, is 7. It's neutral. Okay, so this is a pH scale, and it's actually used to determine the concentrations of hydrogen and hydroxide ions. Okay, this is just the pH scale, again showing the substances and also their pH. For example, Coca-Cola has the pH of 3. Okay, vinegar has a pH of 3, so it has higher hydrogen concentrations than hydroxide ions. So those solutions that have a pH which is greater than 7, we call them alkaline or basic, and the concentration of the hydroxide is actually greater than that of the hydrogen ions. In the same vein, the solutions that have a pH which is less than 7, they are said to be acidic. So two solutions, they will differ in pH by 1 unit, which means that one solution has 10 times the hydrogen concentration of the other. So if the pH only differs by one unit, one pH unit, it means that the concentrations in terms of the hydrogen, it's actually 10 times than the other one. Okay, someone is asking, they want to know the difference between 1.8 times 10 to the power negative 16 and 1.0 times 10 to the power negative 14. Okay, so the 1.0 times 10 to the power negative 14 comes as a result of you multiplying 55.5 by 1.8 times 10 to the power negative 16. When you multiply 1.8 times 10 to the power negative 16 with 55.5, it will give you 1.0 times 10 to the power negative 14. I hope that is clear. Okay, so the pH of an aqueous solution can be approximately be measured by various means, by various indicator dyes, which include the litmus, phenolphthalein, and also phenol red. And these actually undergo color changes. That's why they're able to measure the pH approximately. So the accurate determination of pH in the laboratory can actually be made with a glass electrode that is selectively sensitive to the hydrogen concentration, but not sensitive to the sodium, potassium, and other cations. So you just use a pH meter, and in a pH meter, the signal from such an electrode is actually amplified and to be compared with the signals that will be generated by a solution of accurately known pH. Okay, so when measuring the pH, so pH is actually one of the most important and frequently used procedures in biochemistry. We want to know the pH of blood because we need to know what is normal and what is abnormal. Okay, because if the pH of blood, for example, is slightly, it's actually, if it's way below 7.2, it will, that kind of a blood will be referred to as acidosis, acidotic. Okay, so that would be metabolic acidosis, which is a bad thing. And if the pH is actually more than the 7.2, we are going to say that that particular blood it's basic, and that is very bad because metabolic alkalosis is a very bad thing. So the pH will actually affect the structure and also the activity of biological macromolecules. So the pH will actually affect the structure and how the biomolecules interact with one another and other molecules. For example, the catalytic activity of enzymes is actually strongly dependent on pH. So enzymes can only function at an optimal pH. So we have to know how to measure the pH of blood and also urine. Okay, for example, if we find that the pH of urine is basic, we know that there's something wrong. Okay, because it's slightly acidic. Okay, so to diagnose certain diseases, you're going to use your pH because you know that the normal pH of blood is 7.2. Anything that is not 7.2 is abnormal. So the pH of the blood plasma of people with severe uncontrolled diabetes mellitus, for example, sugar disease, is often below the normal value of 7.4. So the 7.4 actually is the normal. Okay, so in some books you may find 7.3 or 7.2. So for us, 7.4 is the normal. Anything below that, for example, in diabetes mellitus, when you measure the pH of blood, you find that it's actually below 7.4. This condition in medicine is called metabolic acidosis, okay? And it is a very bad thing in these diabetic patients. We actually call it DKA or diabetic ketoacidosis, okay? So in certain other diseases, disease states, the pH of the blood is actually higher than normal. And these conditions in medicine, we say metabolic alkalosis. Okay, now you try to imagine that there are actually some substances that have both acidic and basic properties. Substances like amino acids, okay, proteins, they may exist as acids or bases. Such compounds are called ampholites. So ampholites are actually molecules that contains both the acidic and basic groups. And all of the common amino acids that are found in proteins are actually ampholites. Why? Because they contain a carboxyl group. So Tutor Ferguson is going to teach you about these functional groups, the carboxylic acids and their reactions, the mechanistic things, and all those things. You're going to learn from organic chemistry. Okay, so you should understand that some molecules like the amino acids, they contain both the acidic part and the basic part. So here we can see the carboxyl functional group that acts as an acid, and the amino group, the NH2, which acts as a base. So as free amino acids, each of these amino acids has at least 2 pKa values. Okay, some of them they can even have more pKa values. So therefore, the titration of an ampholyte will generate a more complex plot of the pH versus the concentration. Okay. All right, so that was pH and its determination. Now we cross over to the buffering, to the buffers and also the physiologically important buffers in humans. Any questions concerning the pH and its determination? Anybody with a question? All right, so we can move on. Okay, so we said that ampholytes are molecules or compounds that both the acidic and the basic properties. So they can act as acids or bases. For example, we have amino acids, or the 20 amino acids that you know, they are ampholytes in nature. And even the titration curve of the pH against the concentration is quite complex, as we are going to see next week, because next week, proteins.
As for
Okay,
me, I
so
wanted to ask on the acidosis and alkalosis. I don't know if it's
okay,
alkalosis, yeah. But is it diabetes the only condition which falls below 7.4, or there are more conditions that fall under acidosis? And
Doc,
also,
there are thousands of diseases that will give you the acidosis. Thousands of diseases. You can't even mention them. There are a lot. Okay? It's not just diabetes. There are so
all
many
right.
things that make you to have that acidosis, as you are going to see. So now we go into the last component, which is the buffer system. Okay, so anyone who can define a buffer for us? Anyone willing to define a buffer? Blessing, are you in class?
Yes.
Anyone who can define a buffer for us? Yes, Blessing. You can unmute yourself. Blessing, can you kindly unmute yourself?
Yes. A buffer system is used to attract both acids and bases. I'm not sure, sir.
Okay, fair enough. Anyone else define a buffer?
Uh, it's a solution of a weak acid and a strong conjugate base.
Well, there's mixed up of data here. So a buffer is a solution that resists change in its pH when a small amount of an acid or base is added to it. A solution that resists a change in pH when a small amount of an acid or a base is added to it. Okay, so almost every biological process is pH dependent. Whatever is going on in the cell is pH dependent. So a small change in the pH will actually produce a large change in the rate of that chemical reaction. For example,
Okay.
the enzymes that catalyze cellular reactions and many of the molecules on which they act, they contain ionizable groups with characteristic pKa values. So the protonated form of the amino acid and the carboxyl groups of the amino acid, actually, and also the phosphate groups of the nucleotides, for example, they function as weak acids. So their ionic state depends on the pH of the surrounding medium. Okay, so the cells and also the organisms, they maintain a specific and also constant cytosolic pH. So the pH of the cytoplasm, which keeps the biomolecules in their optimal ionic state. Okay, which is usually a pH of 7. So in multicellular organisms, the pH of the extracellular fluids is also tightly regulated, like for example, the pH of blood. So the constancy of pH is achieved primarily by biological buffers, which are mixtures of weak acids and their conjugate bases. So weak acids and their conjugate bases. Buffers are actually aqueous systems that tend to resist changes in pH when a small amount of an acid or a base is added to it. So for example, we have the bicarbonate buffering system. Okay, so a buffer system will consist of a weak acid, which is the proton donor, and its conjugate base, which is the proton acceptor. Okay, so here, as you can see, we have water and its ionization. Okay, we have acetic acid, which is a weak acid. So acetic acid will undergo ionization. The HAC is the acetic acid. So it will, when it is reacting with the hydroxide ions, you see what is happening. It donates the proton. In this case, it's acting as a proton donor. Okay, but um, when it's reacting with an acidic substance, as you can see here, this hydrogen will be swallowed by the conjugate base, which is this one, as you can see. Okay. Therefore, acetic acid is a buffer. Is actually a buffer. Okay, so it will resist a change in its pH. Okay, so a simple expression of the rates of the pH, the pKa, and also the buffering concentration. So what is going to relate the pH, the pKa, and the concentrations of the buffer, the buffer components is what is called the Henderson-Hasselbalch equation. There's an exam question asking you what the Henderson-Hasselbalch equation is. It asks you what is this thing and where does it apply. Okay, so the Henderson-Hasselbalch equation is very important for understanding the buffering action and also the acid-base balance in the blood and also the tissues of vertebrates. Okay, so for you to understand the acid-base balance that happens in the blood and also the tissues of the vertebrates and animals, like which includes humans, for example. Okay, so this is important because it is going to help you to understand the buffering action that happens in humans. So this equation is simply a useful way of restating the expression for the dissociation constant of an acid. So when you get the dissociation of an acid, the previous acid, the acetic acid, you'll see it's going to give us, it will reach to the Henderson-Hasselbalch equation. We call it the HH equation in biochemistry. So for the dissociation of a weak acid, which is HA, into the H plus and the A, the Henderson-Hasselbalch equation can be derived as follows. You have to know how to derive the Henderson-Hasselbalch equation because they'll ask you to derive it and then you get stuck in the exam. So you start with the Ka, which is the equilibrium constant of this acid, and this is how you express it. First, you're going to solve for the concentration of the hydrogen ions. So as you can see here, the concentration of the hydrogen ions, you make it as the subject of the formula. Then you take the negative log on both sides of the equation. So here on this step, you have done that. And then you're going to substitute where there is the negative log of the hydrogen, you put pH, okay? So when you put pH here, we're going to put pH here. Here we're going to put pKa because the P stands for negative log. Here we are going to put it. We're going to leave it like this anyway because it's what we're going to find. So this is the ratio. So this one is the ratio of the acid over the conjugate base. Okay, so here there is pH, there is pKa, and there's also this ratio of the acid to its conjugate base. Very important. So here we can put a plus, and rearrange these. This goes on top, this comes down. Okay, there's such mathematics. So you can be asked the ratio of the acid to its conjugate base, or the ratio of the conjugate base to its acid. Okay. So what I've written here is this one. So we have rearranged it, okay, so that it has a plus. So what you are seeing here is called the Henderson-Hasselbalch equation. In the exam, you'll be asked to derive this. Just show how you derive the Henderson-Hasselbalch equation. Okay. And so it shows that the pKa of a weak acid is actually equal to the pH of the solution when the concentration of the acid is equal to the concentration of its conjugate base. So in this and this, they have equal concentration. This becomes a 1, and log of 1 is actually 0. So which means at a concentration where this and this are equal, it means that the pH is equal to the pKa of that particular weak acid. Now, the other question in the exam is where do we apply the HH equation? So the HH equation can be applied to calculate the pKa1, to calculate the pH, to calculate the pKa if you have been given the pH and also the molar ratio of the proton donor and the acceptor. Number 2, you can calculate the pH when you have been given the pKa and the molar ratio of the proton donor and the proton acceptor. The last thing is that you can use the Henderson-Hasselbalch equation to calculate the molar ratio of the proton donor and the acceptor when you have been given the pH and the pKa. Just by rearranging the equation, you are going to get the molar ratio. Now, what are the important physiological buffer systems? Before we continue with that, any question on the Henderson-Hasselbalch equation, because it is very much examinable? Any questions?
Yes, Doc, maybe you can just explain again on how to drive it.
Okay, it's a very simple process. So you start with the acid, the HA being dissociated to the H and the conjugate base. So there's this equation which is going to show us the dissociation. Then when you get the Ka of that particular acid, remember it's a weak acid. Okay, so weak acids, they don't completely dissociate in water. So you see that the product is this and this, the reactant is this. Okay, so now here you're going to make the H plus as the subject of the formula. Once you're done with that, you're going to take the minus log on both sides. When you do that, you guys, you know mathematics very well. It will give you this gamma equation, which is here. And this one, you realize that the negative log of this is actually pH. You put your pH here, you put your pKa here, then minus log of the concentration of the acid over the conjugate base. Okay, and here now you can see clear. We don't do this in science. So we put a plus. When you put a plus here, they will swap. The denominator will swap with the numerator, and it will become this. In the exam, for example, if you end here, you'll be given marks will be deducted because we don't write the Henderson-Hasselbalch equation with a minus. There is always a plus here. Okay. I hope that is clear. So when the concentration of this and this, they are equal, it means that the pH is equal to the pKa of that particular acid, because they can ask you to calculate the pH, they can ask you to calculate the pKa, and of course they can ask you to find the molar ratio of the conjugate base over the acid. Okay, so now we move on to the last part, which is the physiologically important buffers in the body fluids and also the tissues. So this is directly linked to the human been now. So the intracellular and the extracellular fluids that are found in the cells of humans, they're actually they have a characteristic and nearly constant pH. So the cytoplasm of most of these cells that you're going to study in anatomy, they actually have high concentration of proteins which contain many amino acids with functional groups that are weak acids or weak bases. The nucleotides such as ATP, which is the vessel energy currency, as well as many low molecular weight metabolites, they contain ionizable groups that can contribute the buffering power to the cytoplasm. So the ATP and also the nucleotides can also contribute to the buffering power that is seen in the cytoplasm. So some highly specialized organelles and also extracellular compartments, they have high concentration of these compounds and that actually contributes to the buffering capacity. So the organic acids, they buffer the vacuoles of plant cells. For example, ammonia buffers urine. So the ammonia is very important because it buffers the urine. So we have two important biologically important buffers, which are the phosphate and the bicarbonate buffer system. Guys, this is examinable. You have to pay much attention to the details. So the phosphate buffer system, which acts in the cytoplasm of all cells, actually consists of the phosphoric acid as a proton donor and the phosphate, the hydrogen phosphate as a proton acceptor. So there's one which is an acceptor, one is a proton donor. Okay, this one with an H2 is a proton donor, it will donate the proton. This one is a proton acceptor. So as you can see, protons are being lost from this to form this, right? So the phosphate buffer system is effective at a pH which is closer to its pKa. So the pKa of the phosphate buffer is actually 6.86. Okay, and it is strongest at that pH which is closer to that. Therefore, it tends to resist the pH changes in the range between 5.9 and 7.9 respectively. Therefore, it is an effective buffer in biological fluids, in mammals, for example, in humans, extracellular fluids, and most of the cytoplasmic compartments, they have a pH in the range of 6.9 to 7.4. So the blood plasma is buffered in part by the bicarbonate buffer system in blood. So in tissues, it's the phosphate buffer. In blood, it's the bicarbonate buffer system. So the bicarbonate buffer system contains the carbonic acid as a proton donor and then the bicarbonate as a proton acceptor. Okay, so this is the equation for the bicarbonate buffer system, just like you saw in the phosphate buffer system. So this one will undergo ionization, as you can see. Its first ionization will still leave the proton here. That's why we refer to this as K1. So there's K1, K2, K1 and K2 for the bicarbonate buffer system. Okay, you already know how to write down the equation for the equilibrium constant. So this buffer system, which is the bicarbonate system, is actually more complex than other conjugate acid-base pairs, but it's complex than other buffering systems. Why? Because most of its components, which includes the carbonic acid, is actually formed from the dissolved carbon dioxide and water in a reversible reaction. So there's a reaction here where carbon dioxide is changing from its gaseous state to its dissolved state. So you have to pay attention to this. There's actually the K3. So K1, K2, and K3. Okay. So the carbon dioxide is a gas under normal conditions, and the concentration of dissolved carbon dioxide is the result of the equilibration with the carbon dioxide of the gas. Okay. So there's a gas phase, as you can see. So there's the gas and the dissolved phase of carbon dioxide. So although many aspects of the cell structure and function they are influenced by pH, it is actually the catalytic process of the enzymes that is especially very sensitive. So enzymes will actually only work at a maximum catalytic activity if the pH is at its optimum, which is called the optimum pH. So on either side of the optimum pH, their catalytic activity will actually decline sharply. If the pH increases or reduces, that particular enzyme won't be able to work, and life depends on enzymes. Therefore, a small change in the pH can actually make a large difference in the rate of some crucial enzyme catalyzed reactions. Okay, so the biological control of the pH of the cells and also the body fluids is therefore very central to understand, and it's very important in all aspects of metabolism and also cellular activities, because even as we start metabolic chemistry, you realize that pH is very important. You have to maintain it, or else that particular enzyme won't be able to survive or to work efficiently. Right. So we have come to the end of the class. As you can see, there is an assignment. So this is the first assignment that you'll be given, and it must be submitted on Friday, 18 hours. So there is an assignment. Just go in there. When you open Google Drive, you'll find the assignment folder. You're going to find this assignment, and it should be submitted on Friday before 18 hours. Okay, so any questions before we close? Any questions?
So these notes that you were just from explaining to us, we'll find them as well on Google Drive.
Yes, you're going to find them together with this video. So this video, the notes, and also the assignment, you're going to find them in their respective folders. When you open the biochemistry folder, you'll see there's so many things. You open the assignment folder to find it.
Thank you.
Okay, any other queries?
Maybe you can tell us how the means in which we are going to submit the assignment.
Oh, okay. So this is how you submit assignments here. You're going to write your answers on a plain paper, then capture photos, convert them to PDF. When you convert them to Okay, so when you convert them to PDF, you're supposed to send the assignment direct in my inbox. Okay, don't send to any other person. You should send the assignment direct to my inbox in form of a PDF, not pictures. So you get nice, clear pictures, then you send to my inbox. You send your PDF to my inbox. Make sure you write your name on that assignment, and also the course that you are writing the assignment for, so that I'll be able to know, even when I'm going to, even when we are marking and reviewing your assignment. Okay, someone is asking. Okay, so yes, these notes, they have actually been used by people who have passed. You can see that Davis, who actually left, was using these same notes. You don't even need the class notes because they are synchronized. These notes are synchronized with the university notes, and you actually just need these notes to pass the exam. Any other questions? Yes. So guys, you just need these notes to pass your exams. So all the notes are actually there from A to Z, just that you can't see them. You can only see them once you have learned a topic.
Any other queries? Okay, so this is exactly 2 hours because our classes will be all 2 hours. No, there's no next class. The next class is tomorrow for basic and applied human anatomy and development. So there's no next class. Just wait for the recording. So it takes time for the Google guys to process the recording. So you're going to find the recorded session on our YouTube channel. You can actually download it, or you can find it on Google Drive. Okay, so Google Drive as the folders, and in the folders you can see the recorded sessions and also the slides. So each recorded session, each topic has its own slides, it has its own recorded class. That's how it is. So everything will have its slides and its recorded session. And there will also be a transcription. But for this class, I was unable to do the transcription because it was going to make other students to leave the class. So in a transcription, this is where we have whatever we are talking about is being transcribed into words, and then we can actually produce a PDF document that we can send to the rest of the group and people can actually see. So assignments are actually mandatory. You have to write these assignments and submit, okay, and submit on time. So submission is before 18 hours Friday, and make sure that you study before you answer this assignment because it just contains exam questions. So there's nothing simple about the assignment. It's just exam questions. Any other queries? If you have a query, please. Loveness, you can unmute yourself and ask.
No, sir, thank you. I don't have any other questions.
Okay. Okay, that's good. Okay, so the recording will be available tomorrow, obviously, because Google has to process the recording.